Greetings from Michigan State University, and welcome to EAB University's 2017 Spring Webinar Series, which is funded by the USDA Forest Service. This is Robin Usborne coming to you from Michigan State University, and along with my EAB University colleagues, Cliff Sadoff from Purdue University and Amy Stone from The Ohio State University, we welcome you to today's webinar, Biology, Ecology, and Management of Ambrosia Beetle Vectors and Their Diseases, being presented by Chris Ranger. Chris is a research entomologist within the USDA-Agricultural Research Service located in Wooster, Ohio. He is also an adjunct assistant professor within the Department of Entomology at The Ohio State University. He obtained his bachelor's degree from Ohio State, his master's degree from Pennsylvania State University, and his PhD from the University of Missouri. Chris has been with the USDA since 2006, and the main focus of his research program is to develop sustainable management practices for tree-infesting ambrosia beetles. Chris has authored over 50 peer-reviewed scientific journal articles and serves on the editorial board of the Agricultural and Forest Entomology and Environmental Entomology publications. Before we get started today, please know that we welcome your comments and questions, so please feel free to type them in the chat feature, which you can find by mousing over the bottom or occasionally the top of your screen. We will make a note of all the questions and we'll have Chris respond to them when his presentation is finished. To keep these free webinars coming, we need your feedback. After the webinar, I will be emailing a link to a short, voluntary, confidential survey that I hope you'll take the time to fill out. If you're one of the first 10 people to fill out the survey, we will be sending you an EAB goodie bag. For those of you wanting CEUs, if you would like a certificate indicating that you participated in today's live webinar, complete the survey and send an email message to Amy Stone at stone.91@osu.edu. I will also put these instructions in the email that I send you. Certificates will be mailed to you within a week of today's program. This webinar is being recorded and will be available for viewing soon at www.emerald-borer.info. You will also find the recordings for all our previous EAB University's webinars there. Thank you for attending today. And Chris, please unmute your microphone, share your screen with your presentation, and we'll get started.
Let's see.
All right.
Robin, can you?
There you go.
Yes, you can. Okay, great. Okay, let's go ahead and get started then. Thanks to Cliff and Robin and Amy for the opportunity. So basically, bark and ambrosia beetles are among the most destructive pests of trees in horticultural and forested landscapes. They are commonly intercepted at ports of entry because of their wood-boring behavior, and that can involve live plant material, plant products, and wood packaging material. As of 2010, there are 58 species of bark and ambrosia beetles that are established in the United States. Now, in terms of bark beetles, they feed on actual plant tissue, including the phloem, and that also includes fungus-infected tissues. Whereas ambrosia beetles are actually feeding on a symbiotic fungus that they farm within the host tree. Now, in terms of a regionally important, economically important bark beetle that I want to mention, that some of you may have heard of, is the walnut twig beetle. And that is attacking black walnut and other juglans species and transmitting thousand cankers disease. And so for those of you that are interested in learning more about this destructive bark beetle, I'd encourage you to take a look at some of the work that Matt Ginzel with Purdue has been doing. But today I'm going to be focusing on some exotic ambrosia beetles. And two species in particular are of significance in ornamental nurseries and tree fruit orchards, but also in forested settings. And that is the black stem borer, Xylosandrus germanus, and the granulate ambrosia beetle, Xylosandrus crassiculatus. Now, the black stem borer is currently established in 32 of 50 states, including several provinces in Canada, and also throughout much of Europe. The granulate ambrosia beetle is currently found or established in 29 of 50 states. It has less of a distribution in Europe, but also is found in Central America, the Caribbean, parts of Africa, and Oceania. And so both these species, I should mention, are native to southeastern Asia. The black stem borer was first detected back in the 30s in North America in New York, and the granulate ambrosia beetle was first detected in 1974 in South Carolina. Now, both for these species and ambrosia beetles in general, the males are flightless, so it's the females that are making the host selection choice, and therefore it's the female that is most important for identification purposes. Now, as I mentioned, they have a wood-boring behavior, so it's the female that actually will tunnel into the host tree. In terms of symptoms, they create a relatively narrow hole typically in the stems, but also sometimes the branches. But for these two species, it's really the stems. It's a relatively small hole, and as a result, the attacks are not always easily detected. But there are some key traits or symptoms. One of those are sawdust toothpicks that stick out perpendicularly from the stem of the tree. And that is the result of the tunneling of the female. So as she's tunneling into the tree, she's pushing out this basically sawdust material that just gets packed together. But once it rains or if there's heavy winds, the sawdust toothpicks do not last very long. So it's kind of a temporary symptom. Defensive sap production is also a key characteristic of ambrosia beetle attacks that are often seen during the spring months in terms of this defensive sap production. And this sap production can be quite extensive to the point where it can result in opportunistic organisms then taking hold. And so obviously from an ornamental tree perspective, it can really reduce the aesthetic properties of that tree. Now, the females are creating a series of tunnels and galleries deep within the tree. And so they're going well beyond the phloem and tunneling deep into the heartwood. And so the extent to which they're tunneling really depends on the species of the tree, but you can see several different examples here. So, as I mentioned, these insects are fungus farmers, and the females have a pouch called a mycangium that's located in the prothorax. So if you were to slice the beetle in half longitudinally, you would see this pouch called a mycangium, and contained within that are spores of their symbiotic fungus. So once the female tunnels into the tree, she then creates these galleries and inoculates the tree, inoculates the tunnels and the galleries with their fungal symbiont. Now, it's important to note that the females do not begin laying eggs until the fungal symbiont is actually growing in the galleries. So we feel that that's an important weak point in their biology that we're hoping to exploit for management purposes. But in terms of the fungal symbionts, they're Ambrosiella grossmaniae is the fungal symbiont for Zalisandrus germanus, and Ambrosiella roperi is the fungal symbiont for Zalisandrus crassiculatus. Now, both of these species and Ambrosiella in general are not considered to be plant pathogens, and so that's an important point that I want to make. However, these ambrosia beetles are associated with a variety of secondary microorganisms, some of which are known to be pathogenic, and that includes a number of bacteria, yeasts, and fungi that have been isolated from the galleries. And it's possible that spores can be carried on the surface of the beetle, for instance, the cuticle, that are then quote unquote accidentally introduced into the tree, or as a result of an open wound being created in the tree, you could then have these opportunistic microorganisms that would take hold. Now, it's also worth noting that laurel wilt disease, which has been killing red bay trees in the southeastern United States, that has been isolated from crassiculatus, and it has also been transmitted under controlled conditions to red bay and avocado. So that's certainly of concern and important to understand the biology and ecology of these generalist ambrosia beetles. So in terms of wilting and dieback, this is an additional symptom that we see. And for instance, you can see on this magnolia here, there was extensive dieback and basal sprouts that then were produced, and also some wilting associated with attacks on dogwood. But it's the basis for this wilting and dieback is really not well understood. It could be secondary pathogens. There's also some evidence that perhaps the symbiotic fungus is clogging up the vascular tissue, or perhaps there's a plant hypersensitive response where it's essentially over-responding or overreacting to the presence of the fungus, or perhaps some other pathogens. Now, in terms of their seasonal flight activity, basically these ambrosia beetles are overwintering as adults within trees. And so in the spring months, they're then emerging from those overwintering sites and leaving the woodlots that are neighboring ornamental nurseries and tree fruit or orchards. And so basically we see, in terms of trap captures, we see the largest number of trap captures occurring in the spring months. And then a first generation is then detected in the middle of the summer. And it's somewhat hard to tell on this figure, but there are beetles that on occasion that are caught or trapped as part of the second generation in the fall months. Now, the majority of attacks generally occur during the spring months in response to these overwintering adults. Now, in terms of the influence of temperature on flight activity, we have seen that flight activity dramatically increases when there are 2 to 3 consecutive days above 70 degrees Fahrenheit. In other words, so during spring months, before it reaches 70 degrees, it's important to keep an eye out for that forecast, because we see that, again, the flight activity really increases dramatically following those 2 to 3 days of 70 degrees. In terms of first flight for Zalisandrus germanus and Zalisandrus crassiculatus, basically it's about February to March in the southern and southeastern United States, and March to April for parts of the Midwest and the Mid-Atlantic states. But so those are really the first captures, and then at least in Ohio, in terms of peak flight, that generally would occur in April in the Mid-Atlantic states, and then May in in the Midwest. Now, I wanted to mention that eastern redbud is the flowering of eastern redbud does often coincide with a first initial flight of Ambrosia beetles. So as I mentioned, these two species are overwintering in trees in woodlots. And during the spring months in particular, they're then leaving those woodlots and flying into the landscape. Now, in terms of their preference for trees, ambrosia beetles have essentially, depending on the species, they have a broad host range or a narrow host range. And they also have a certain preference for host quality, and that can range from a healthy tree to a weakened tree to a dead tree. And so depending on the species, the host range and host quality is going to vary. In terms of black stem borers, Alisandrus germanus, they can attack over 200 species of trees. The granulate ambrosia beetle can attack over 120 species of trees. They generally prefer deciduous trees over coniferous trees, and they also prefer thin-barked over thick-barked trees. Now, they are traditionally viewed as secondary pests of dying or dead trees, but there have also been reports of attacks on apparently healthy trees too. And so this concept of apparently healthy has been important to us to understand, because from a grower's perspective, they felt like they really wanted to understand what was the basis for beetles attacking trees that they considered to be apparently healthy. Essentially, they woke up one day and they had experienced large numbers of attacks on trees that they thought were apparently healthy. So to us, that's been an important concept to try to understand. So what we found is that despite a broad host range, despite an ability to attack over 120 to 200 species of trees, what we find is that only certain species and certain individuals are attacked within a given location and a given year. So for instance, what you see here are two plots from two separate nurseries. And the black dots represent trees that were not attacked, and the red dots represent trees that were attacked. And the larger the red circle, the more attacks there were on that individual tree. So basically what we found is that there was not a random distribution of attacked trees. So in other words, despite this capability to attack several hundred species of trees, they were targeting certain individuals. Now, in terms of their host location behavior, obviously sight is going to be an important part, the visual aspect in terms of a silhouette of a tree. But olfactory cues are also important, and in other words, the scent of a tree. Now, trees are attacked in the presence of ethanol, and this is an important concept to understand. So we did an experiment whereby we baited trees with ethanol and then also left trees right next to those baited trees that were unbaited. So basically, as you can see here, we had about an average of 15 attacks per tree on the baited trees, but no attacks on the non-baited trees. But you can see here that the attacks ceased. Well, that was actually the point at which we purposefully removed the ethanol bait from those trees. So once the ethanol bait was removed, the attacks ceased. So beetles no longer view those trees as attractive. We also see a positive dose response with ethanol. So we used a pressurized injection system to inject solutions of ethanol into trees and then monitor the number of attacks on those trees. And so basically with an increasing concentration of ethanol that we injected that was then subsequently emitted from the bark of those trees, in this case magnolia, we saw a positive dose response. However, no attacks on neighboring trees that were injected with water. Now, we did additional experiments looking at how efficient beetles are at finding trees that are emitting ethanol. So we did an experiment whereby we had a tree that was injected with ethanol, and then trees that were not injected with ethanol positioned at varying distances from that tree that was injected with ethanol. And a number of those trees also had sticky material that was applied to the stem to look at landing rates of ambrosia beetles. So when we did that, we found that there were no attacks on any of the trees, regardless of the proximity of the tree that was emitting ethanol. So in other words, no attacks on trees that were not emitting ethanol. And in fact, beetles rarely landed on those neighboring trees that were not emitting ethanol. We didn't catch any beetles on neighboring trees in 2011 and very few beetles in 2012. But in fact, on the trees that were emitting ethanol, we had an average of 17 attacks per tree in 2011 and 26 attacks in 2012. So in other words, beetles have a very efficient capability of locating trees that are emitting ethanol. And this in part explains why we find clusters of trees attacked within nurseries. In other words, why they're attacking certain individuals and certain species within a given year and location. Now, why ethanol? Well, ethanol is emitted in response to a variety of physiological stressors, as you can see here. What we're finding is that flooding, drought, frost injury, and freezing injury are really the key factors that within nurseries and tree fruit orchards that are contributing to attacks or contributing to predisposing trees to attack. Now, a really important concept to understand here is that ethanol can be induced within 1 to 2 days of stress being imposed, and we've seen that for flood stress and also frost injury. And so in other words, you can have ethanol being very rapidly produced and then emitted from those trees. So while those trees to us are asymptomatic or quote unquote apparently healthy, they are still physiologically stressed and emitting ethanol. And beetles are then very efficiently finding those trees that are emitting ethanol and specifically attacking those trees. In terms of waterlogging or flood stress, basically the physiological basis for that is when a tree is deprived, when the roots are deprived of oxygen, the roots are going to switch over from aerobic respiration to anaerobic respiration. And a byproduct of that is ethanol. And that ethanol then travels up from the roots and is emitted from both the stems and the leaves, but we're finding actually the majority of it is emitted from the stems. Now, we noticed that there were a few instances of extreme precipitation events that preceded attacks, and this is what was part of our initial exposure to try to understand the host selection behavior of these beetles. So for instance, a grower in northeastern Ohio reported attacks on dogwoods, only dogwoods within one row, and neighboring species that certainly are on the host range of these ambrosia beetles were not attacked. And so when we talked with the grower about the history of those trees and if they were perhaps stressed in any way, he responded that, well, in fact, these dogwoods were in standing water for 14 days. That was following a severe flooding event that took place in July of 2006. So more than likely, those trees were attacked probably either right away or in the fall. And you can see here the attacks that occurred on those trees. Now, in 2011, we also again saw extreme precipitation events preceded attacks. And there was record precipitation in northeastern Ohio in 2011, and a grower reported symptoms and attacks on dogwoods. And the symptoms included this extreme level of dieback that you see here on these dogwoods, but neighboring trees were not attacked. So we looked at 166 of these trees. And as you can see here, 99% of these trees exhibited dieback, but only 70% of the trees were actually, of these symptomatic trees were attacked. So in other words, these ambrosia beetles were acting as bioindicators of tree stress. So they weren't responsible for the extreme dieback that you see here. For instance, this particular tree had diebacks but no attacks. And when we looked at the vascular tissue, you can see this necrotic zone moving up from the roots. And when we looked further at these trees and talked with the grower, it became evident that these dogwoods were planted in the lowest lying area of the farm. It was very poor drainage on top of the fact that it was a record precipitation event in that region. And in addition to that poor drainage, it also appeared as though planting depth could be an issue, as you can see here, which for dogwoods is not desirable. So we then, as a result of these observations, we conducted controlled experiments. And we used dogwoods because of their intolerance to flooding. And what we found is that actually more ambrosia beetles were attracted to flood-stressed dogwoods compared to non-flooded dogwoods. And on top of that, only the flood-stressed dogwoods were attacked, whereas the non-flooded dogwoods were not attacked. And we also were able to measure and detect ethanol being emitted from the stems of flooded trees, but not from the non-flooded trees. So we then took this a step further and decided to give beetles a choice of flood-intolerant versus tolerant trees. And so what we did was we selected trees that were essentially intolerant to flooding, moderate tolerant, and tolerant. And then again, gave beetles a choice among flooded and non-flooded representatives of each of those species. And the reason for that is that generally speaking, a tree that's intolerant of flooding will often switch to anaerobic respiration faster than a tolerant tree. And so as a result, we predicted that we should see higher ambrosia beetle attacks on those intolerant trees and also higher ethanol levels associated with those intolerant trees. So we did this experiment in Ohio, which essentially has the predominant population, the highest populations well, in terms of the species, Zalastandus crassus germanus, is the most dominant species, whereas in the mid-Atlantic states, Zalastandus crassus culus is the most abundant species. So we conducted this experiment in Ohio and Virginia, and these are the Ohio data here. And basically on the x-axis, you have days after initiating flood stress. So within 3 days of initiating flooding, we had started to have attacks on those trees. And on the y-axis, you have cumulative attacks. And so basically what we found was that, in fact, dogwood and Japanese snowbell, Styrox, and redbuds, which are known to be essentially intolerant to flooding or moderately tolerant, had significantly more attacks than those trees that were tolerant, for instance, river birch and swamp oak. And on top of that, no attacks on any of the non-flooded trees. When we analyzed those trees for ethanol, after 3 days, in fact, we found that there were higher levels of ethanol associated with the intolerant species compared to the tolerant species. So again, beetles were very efficiently locating trees that were physiologically stressed and emitting the highest concentrations of ethanol. And ethanol was not detected in the non-flooded trees. Now, we know that these beetles are opportunistic colonizers of stressed trees, and we were wondering about their ability to essentially spill over from a stressed tree onto a neighboring healthy tree. And that's kind of a scenario that we see often with bark beetles in terms of large-scale outbreaks. And so we were in the process of conducting an experiment, and where we had paired trees that were flooded and non-flooded. And basically the numbers underneath these trees represent the number of attacks on those trees. And so as you can see over here, we found that on one of the non-flooded trees, we had one attack, and that was paired with a tree that had 338 attacks. And then another tree flooded that had 337 attacks, had 8 attacks on the neighboring non-flooded tree. And that was really unusual for us in terms of experiments that we've conducted over several years in several locations. It was really unusual to see attacks on neighboring non-flooded trees. But when we cut into those trees, we found that in fact the attacks on the non-flooded trees were superficial and absent of beetles. So in other words, they just superficially tunneled in and did not colonize that tree. And again, the point to remember is that the fungal symbiont needs to start growing in that tree before the female will begin laying eggs. So we took this a step further and actually caged beetles with no choice to flooded and non-flooded trees. And when we did that, we found that there was significantly more sawdust that had piled up in the chamber of the in the chamber of the beetles that were caged to flooded trees. And we also found that there were significantly more living founduses, eggs, larvae, pupae, and also the presence of the fungal symbiont in galleries that were created in the flooded trees, whereas we had no living founduses, no eggs, no larvae, no pupae, and no fungal symbiont associated with the beetles that were caged with no choice to the non-flooded trees. So in other words, they're very poor colonizers of healthy trees. So as a result of this association with flood stress, we worked with Steve Frank at NC State to develop an integrated pest management tactic. And basically what Steve found was that a media moisture threshold of less than 50% is really ideal for flood-intolerant species. However, during peak flight, ornamental nurseries are often operating at 50 to 90% moisture media. So that's something to be aware of in terms of trying to minimize attacks on flood-intolerant trees. Now, it's very important to consider that flood stress is, again, not the only basis for attacks in nurseries or tree fruit orchards or forested settings. And some research that was conducted by Jean-Claude Gregoire's group over in Belgium involving Zalisandrus germanus, they in fact found that attacks by Zalisandrus germanus and other opportunistic Ambrosia beetles on large stands of beech trees were preceded by extreme frost events. And they conducted additional experiments that demonstrated that frost-injured tissues were more attractive to Ambrosia beetles than neighboring tissues that were not injured. Now, in terms of some field observations, we have seen that frost injury has preceded attacks, in particular in 2012. We had mild winter temperatures in Ohio in 2011-2012, in that winter. And in fact, April ended up being cooler than March. There were 5 frost events that occurred in April. And following those frost events, there were attacks on intolerant species. For instance, this Kentucky yellowwood here tried to leaf out twice and was zapped both times, and then subsequently Ambrosia beetles attacked that particular tree. And this was a large specimen tree that was growing in a homeowner's yard. So we've conducted some controlled experiments looking at the ability of frost injury of Styrox japonicus, Japanese snowbell. And basically, we found that, in fact, frost-injured trees were preferentially attacked, and beetles, again, started attacking those trees within 1 day. of frost injury being imposed, and essentially peaked around 4 days. We looked at ethanol levels associated with those trees that were frost injured, and in fact, we had some ethanol being induced after 1 day, and it peaked around 4 days, which again coincided with peak number of attacks. And no ethanol emitted from the non-frost injured trees. Now, currently, we are in winter of 2016-2017 into spring 2017. We are currently a month ahead of schedule. And of course, April frosts would not be unusual. As of yesterday, we were at 82 growing degree days, and this is from the Ohio State phenology calendar. And so basically, I just wanted to just point out that in March 2012, again, which was a very mild winter, and we had those spring, April freezes, there was significant losses due to ambrosia beetles. So if we have a frost event in April, it could be problematic for ambrosia beetles. Now, I also wanted to mention that freezing injury has preceded attacks, and this was associated with the polar vortex winters that we had really in 2014 and 2015. A lot of record-breaking low temperatures throughout the Midwest and the Mid-Atlantic states and the Southeast. And in Ohio in particular, we saw extensive ambrosia beetle attacks on a number of ornamental trees. For instance, you can see this cracking here of the stem and ambrosia beetle attacks that occurred on that particular tree. And I should mention that in 2014-15, there were extensive ambrosia beetle attacks that occurred on fruit trees throughout the eastern United States. And that was really the first indications of ambrosia beetle issues within a long time. Really the first indications of reports of ambrosia beetle attacks were on tree fruits to the point where they felt it was essentially a new pest. But when in fact they've been established for a long period of time, and in fact, Zalsandus crassiculatus was first identified or detected from peach trees in South Carolina in 1974. So it appears as though the low temperature injury that occurred resulted in attacks on tree fruits. So in terms of monitoring flight activity for ambrosia beetles, obviously ethanol is, as you can imagine, the most attractive volatile attractant that we have for these insects. Now, the ethanol lures can be purchased or made, but I strongly recommend purchasing them just because of the if you make the dispenser, the challenge with that is it can evaporate quite quickly to the point where you really have to refill or replenish the ethanol levels after 2 to 3 days. Now, the important thing is if you do decide to make your own lures, you have to make sure that you use ethyl alcohol, not isopropyl alcohol. In terms of the traps can be purchased or homemade. Certainly making them at home is easy and inexpensive, and you should include some soapy water or low tox antifreeze, but really soapy water is sufficient as a preservative and a killing agent. And it's also very important to place these traps low to the ground because these ambrosia beetles are low flyers, and also place them within or near a woodlot because that's where you're going to be that's where beetles are going to be leaving in terms of moving into a farm. Now, you can also monitor Flight activity using a ethanol soaked bolt. This was from a colleague of mine, Pete Schultz at Virginia Tech, where they soaked a bolt in 95% ethanol for 20. Oh, excuse me. Well, they cored it. So they drilled out the center of the stem and then plugged it, and it filled it with ethanol and then plugged it. But you can essentially achieve the same thing by soaking the bolt in ethanol for 24 hours. You can also flood stress an intolerant tree like a dogwood using a pot-and-pot system. But the important thing is just to make sure that you're using, make sure that you're maintaining standing water within this pot in order to allow for the ethanol levels to build up. And the nice thing about this is that, for instance, soaking in ethanol, the nice thing is that you can really quickly see when ambrosia beetles are active. So in terms of management tactics, maintaining tree health is really absolutely the first step. And also, in line with that, it's very important to recognize the significance of abiotic stressors. And we certainly don't have all the answers as far as all the stressors that can occur and predispose trees to attack. But that's where it really is important to interact with growers and get feedback in terms of what they are seeing and what might have predisposed trees. But certainly flooding, frost injury, and winter injury are what we are seeing as reoccurring issues. Now, in terms of preventive chemical controls, permethrin and bifenthrin-based insecticides are among the most effective, but they're definitely not 100% effective at protecting trees, especially if those trees are emitting ethanol. If they're not emitting ethanol, the beetles won't attack them, and there's no need to apply an insecticide. Systemics are not effective, and treatments should be applied prior to peak flight. So that's why it's important to get traps out. And then on top of that, thorough coverage of the trunk is really key. As you can see here, the majority of attacks occurred on the trunks compared to the branches. Now, the reason why it's so important to maintain tree health is that insecticides reduce but do not prevent attacks on stressed trees. For instance, here's attacks on a flood-stressed tree. Here's attacks on a flood-stressed tree that was treated with permethrin, and then no attacks on the non-flooded trees. Now, we do see that permethrin pre-treatment also, in addition to reducing attacks, it can also reduce the colonization success. In other words, we saw that galleries with fungal growth were significantly lower, and also galleries with eggs were significantly lower on trees that were pre-treated with permethrin compared to untreated trees. Now, from an ornamental standpoint, that's still problematic because really ornamental producers have essentially a zero threshold for ambrosia beetle attacks, but perhaps with other commodities it could be useful to disrupt the colonization success. In terms of the fungicide pre-treatment, this has come up in interactions with growers. Basically, we did not find these two fungicides to reduce attacks, but we did see that they could potentially disrupt fungal growth or fungal establishment of the fungal symbiont, and also, as a result, disrupt the number of galleries with eggs. So in terms of Generalist ambrosia beetles, in terms of some conclusions, trees in the early stages of stress are rapidly attacked. And while those trees may appear asymptomatic or apparently healthy, they're actually emitting stress-induced ethanol. And so that's really the most important cue for these insects. And so understanding what factors induce ethanol is key. And again, they are poor colonizers of healthy trees. And flooding, poor drainage, frost injury, and low temperature stress are among the key scenarios here. In terms of a management plan, again, maintaining tree health, consider physiological stressors, monitor spring flight activity using ethanol beta traps, preventive treatments using Permethrin or Bifenthrin, depending on, of course, what is on the label for your particular commodity. And you can use heavily infested trees as trap trees for a couple of weeks after the attacks are first detected. And then for those heavily infested trees, you would want to cut those down and essentially burn or chip them to a very fine chip. And now I should mention that it's not always a death sentence, that trees with a light infestation can recover, but in an ornamental scenario, it's unlikely that those trees would be marketable. So again, it's really up to the grower's discretion. They may decide to, in other words, keep those trees around for another couple of years, and the attacks can heal. So. And in terms of acknowledgments, I just would especially like to note the Floriculture Nursery Research Initiative as a funding source, along with the Horticultural Research Institute. And I have a number of collaborators listed here that I'd like to acknowledge, and of course, technical support. And so with that, I'd like to thank you for your attention, and I'd be happy to answer any questions.
Well, thank you, Chris. I appreciate the discussion, and it's, enlightening all of us. I do have a couple of questions from our audience. Cliff Sadoff says, we have already had a couple of days of temperatures over 70 degrees Fahrenheit, but we have not yet had red buds bloom. In oddly warm winters like this one, would you expect the trees to still be a reliable indicator?
Okay, well, I guess the jury's still out because we've been monitoring. This is in conjunction with and really being led by Jason Oliver with Tennessee State University, and he's been monitoring plant phenology and ambrosia beetle emergence over the past, I think probably a good 6 to 7 years now. And so over the past several years, he's been adamant about the relationship between red buds and ambrosia beetles. And so this year, it'll be very interesting to see how things hold up. Now, in the past, in terms of the 70-degree temperatures, again, in the past, that's been a pretty good indicator. Now, with the way things are rapidly progressing this year, it'll be interesting to see how they play out. But basically, I mean, the scenario is that these ambrosia beetles are overwintering as adults within trees. So in other words, they don't need to accumulate a certain number of degree days in order to develop into the adult stage. They're already an adult. So really what they're looking for are temperatures that are warm enough, presumably, for them to begin flight. And so it'll be interesting to see how things shape up. But for me, the biggest concern at this point is the potential for frost injury.
Thank you. And Cliff has another question. So are ambrosia beetle increases another consequence of climate change?
Well, I would say that they're definitely a bioindicator of tree stress. And we do know a number of extreme climatic events have preceded ambrosia beetle attacks. And so I would definitely be concerned in terms of the ability of extreme climatic events to contribute to ambrosia beetle attacks within a variety of commodities. And so the challenge is to essentially not only predict that, but also to essentially manage trees to make sure that they're in an ideal state of vigor.
Okay. We have another one. Would you recommend the application Vasos? Okay, I'm going to screw this up.
Azoxystrobin along
Along.
with Permethrin as a therapeutic. Okay, so yeah, that's a great question, and basically what I think that this is alluding to is the concept of a rescue treatment, and this has come up. In other words, in terms of if trees are already infested, uh, is there a rescue treatment for ambrosia beetles, but also in terms of preventive applications. So in terms of, I'll start with the rescue treatment first. We have conducted some experiments and involving trees that were infested, and then applied fungicides and insecticides to those trees that were infested to again try to gauge if we can essentially disrupt those colonies within those trees. And we need to repeat these experiments, but we do have some data that does indicate that in fact the while the fungicide treatments were not killing beetles, or disrupting the colony, we did find that permethrin treatment on an infested tree did help reduce those populations within the tree. In terms of preventive applications, I don't think so we do see that preventive applications of azoxystrobin can again reduce colonization success. So they're not going to disrupt or reduce the number of attacks, but we did see a reduction in the establishment of their fungal symbiont, and then of course in the number of eggs that were produced, because again, if the fungus is disrupted, then females are not going to begin laying eggs. So we did see that preventive applications of the fungicide, while it didn't reduce attacks, it did disrupt the colonization success. Now, so that could be taken as it could be viewed that perhaps a preventive fungicide application could be used. However, I think on a economical basis, that it's probably not going to be economically feasible to do that. But if it's a tree that's known to be that has a known injury, for instance, frost injury, and it's maybe, for instance, a specimen tree, then I would say that it probably might not hurt for someone to try that. But again, we're still trying to generate some additional data on that.
Okay, thank you very much, Chris.
Thank you.
I am not seeing any more questions at this point, but I will make sure that your contact information is in the email that I send out.
Great.
And other than that, I want to thank you again for all your time and effort to put this together and share it with us on Emerald Ash Borer University. I'm going to be ending the meeting, and unless you have any other comments.
Well, I just would like to thank you. Thank you for the opportunity, and I really appreciate it.
Thank you.